In this module, we will discuss enzyme catalysis. Enzymes are complex biomolecules, typically proteins, that serve as catalysts in a wide range of chemical reactions. These biological catalysts are integral to nearly all metabolic processes in cells, including our bodies. Nature has developed these specific catalysts to facilitate countless biological reactions essential for organism survival. Enzymes are well known for their reaction specificity. They are characterized by high selectivity towards the production of specific products, often accompanied by high activity. Additionally, enzyme-catalyzed reactions typically occur under mild conditions, contrasting with many non-biological chemical reactions that may require harsher conditions. In enzyme catalysis, the reactant is referred to as the substrate. Enzymes contain a specific region known as the active site, which is responsible for their catalytic activity. The active site of an enzyme is specifically designed to bind substrates, facilitating their conversion into products. The structure of the active site is specific to the reaction that it catalyzes, with chemical groups in the substrate attached to chemical groups in the active site primarily by hydrogen bonding, electrostatic forces, and van der Waals interactions. Enzyme catalysis is characterized by a two-step reaction mechanism. The first step involves the binding of the enzyme to its substrate, forming an intermediate complex. This complex is appropriately named the enzyme-substrate complex. The second step is the conversion of the substrate into reaction products and the decay of the complex leading to the release of the products. After the products are released, the enzyme's active site returns to its original state, unaltered and ready for another reaction cycle. Such a mechanism ensures that all enzymes, like all catalysts, do not undergo any net chemical change during the reaction process. This allows them to catalyze multiple reaction cycles efficiently. Experimental studies in enzyme kinetics typically focus on monitoring the initial rate of product formation in solutions where the enzyme concentration is extremely low. This approach is based on the remarkable efficiency of enzymes as catalysts. Even when present in concentrations several orders of magnitude lower than their substrates, enzymes can significantly accelerate reaction rates. The kinetic mechanism of enzyme catalysis is effectively described using the Michaelis-Menten mechanism. This model provides a quantitative framework to understand how enzymes facilitate chemical reactions. The Michaelis-Menten mechanism is visually represented in this figure and can be described by the following reaction scheme. In this scheme, E represents the enzyme, S is the substrate, ES denotes the enzyme-substrate complex, P is the product formed. K1 is the rate constant for the formation of the ES complex. K-1 is the rate constant for the dissociation of ES back to E and S. K2 is the rate constant for the conversion of ES into the product P. According to the Michaelis-Menten mechanism, the enzyme initially binds to the substrate to form the enzyme-substrate complex. This complex can either dissociate back into the enzyme and substrate or proceed to form the product, releasing the enzyme in its original state. The Michaelis-Menten mechanism highlights the dynamic nature of enzyme catalysis. It emphasizes the formation of an enzyme-substrate intermediate complex as a critical step in the catalytic process, followed by its conversion to the product and the regeneration of the enzyme. The Michaelis-Menten equation, derived from the Michaelis-Menten mechanism, is widely employed to analyze enzyme kinetics. The rate of product formation in an enzyme-catalyzed reaction V is a key aspect of the Michaelis-Menten mechanism. It can be mathematically expressed as V equals the derivative of the concentration of P with respect to time and equals K2 times the concentration of the enzyme substrate complex ES. The strategy for analyzing the rate of product formation is centered on finding an expression for the concentration of the enzyme substrate complex ES. The derivation utilizes the steady-state approximation for the concentration of ES. It assumes that the formation and breakdown of ES reach a steady state with the total rate of formation of ES equal to the total rate of its breakdown. The rate of change of the concentration of ES over time equals K1 times the concentration of the enzyme E times the concentration of the substrate S minus K minus 1 times the concentration of ES minus K2 times the concentration of ES. According to the steady-state approximation, this expression approximately equals 0. From the previous equation, it follows that K1 times the concentration of E times the concentration of S equals the sum of K minus 1 and K2 times the concentration of ES. Therefore, the concentration of the enzyme substrate complex ES is given by K1 times the concentration of E times the concentration of S divided by the sum of K minus 1 and K2. To simplify the expression for the concentration of the enzyme substrate complex, we introduce the Michaelis constant, denoted as Km. It is defined as follows: Km equals the sum of K minus 1 and K2 divided by K1. With this definition, the concentration of ES can be expressed as the concentration of E times the concentration of S divided by Km. Note that the Michaelis constant has the dimension of molar concentration. Next, we express the rate law in terms of the total concentration of enzyme and the initial concentration of substrate. The enzyme is either in its free form or bound to the substrate. It follows that the total concentration of the enzyme is given by the concentration of E0 equals the concentration of E plus the concentration of ES. Therefore, the concentration of free enzyme E equals the concentration of E0 minus the concentration of ES. Since we are interested in the initial rate of product formation and the substrate is typically in large excess relative to the enzyme, the free substrate concentration at the beginning of the reaction is approximately equal to the initial substrate concentration. Using these substitutions for the concentration of E and S, the steady-state expression for the concentration of ES leads to the following equation. Km Km times the concentration of Es equals the concentration of E0 minus the concentration of Es times the concentration S0. After rearrangement, we get the concentration of E0 times the concentration of S0 equals Km times the concentration of Es plus the concentration of Es times the concentration of S0. Finally, we arrive at the concentration of Es equals the concentration of E0 times the concentration of S0 divided by Km plus the concentration of S0. This also equals the concentration of E0 divided by 1 plus Km over the concentrations of S0. The expression for the concentration of the enzyme-substrate complex can now be substituted into the equation for the rate of product formation to give the Michaelis-Menten equation. V equals K2 times the concentration of E0 times the concentration of S0 divided by Km plus the concentration of S0, which also equals K2 times the concentration of E0 divided by 1 plus Km over the concentration of S0. When the substrate concentration is much greater than the Michaelis constant, the concentration of the enzyme-substrate complex equals the total concentration of the enzyme. It indicates that all enzyme molecules are bound to substrate molecules as enzyme-substrate complexes. Consequently, the rate of product formation reaches a maximum value, Vmax. This is indicated by the following expression for the rate. V equals Vmax, that is equal to K2 times the concentration of E0. As a result, the product formation rate becomes independent of the substrate concentration. Substituting this definition of Vmax into the Michaelis-Menten equation gives the following alternative expression for it. V equals Vmax times the concentration of S0 divided by Km plus the concentration of S0, which equals Vmax divided by 1 plus Km over the concentration of S0. On the other hand, when the substrate concentration is much less than the Michaelis constant, the Michaelis-Menten equation simplifies to V equals K2 over Km times the concentration of E0 times the concentration of S0, which equals Vmax over Km times the concentrations of S0. For a given total concentration of enzyme, when the substrate concentration is low, the rate of product formation is proportional to the substrate concentration. Therefore, the reaction order in the Michaelis-Menten mechanism changes with the substrate concentration. When the substrate concentration is low, the reaction is first order with respect to it. When the substrate concentration is high, the reaction is zeroth order. This figure illustrates the variation of the rate of the enzyme-catalyzed reaction with the substrate concentration, as described by the Michaelis-Menten mechanism. This graph demonstrates how the reaction rate approaches a maximum rate for large substrate concentrations. By analyzing the Michaelis-Menten equation further, we can ascertain that the Michaelis constant equals the substrate concentration at which the reaction rate is half of its maximum value. Therefore, if the maximum rate is known with certainty, Km can be determined by examining the reaction rate as a function of the substrate concentration. By identifying the point where the reaction rate is half of its maximum value on this curve, the corresponding substrate concentration gives the value of Km. Qualitatively, the lower the value of Km, the higher the enzyme's affinity for the substrate. A lower Km value indicates that the enzyme can achieve half of its maximum reaction rate at a lower substrate concentration, suggesting a strong interaction between the enzyme and the substrate. Conversely, a higher Km value would imply that a greater substrate concentration is needed to reach half of the maximum rate, indicating a lower affinity of the enzyme for the substrate. A valuable tool for the analysis of enzyme kinetics is the Lineweaver-Burke equation. It is derived by taking the reciprocal of both sides of the Michaelis-Menten equation and is expressed as follows. The reciprocal of the rate V equals the reciprocal of the maximum rate Vmax plus the Michaelis constant Km divided by the maximum rate Vmax times the reciprocal of the initial substrate concentration. This equation forms the basis of the Lineweaver-Burke plot, a graphical representation used to analyze enzyme-catalyzed reactions. In this plot, the inverse of the reaction rate is plotted against the inverse of the substrate concentration. For a reaction that follows the Michaelis-Menten mechanism, this plot should yield a straight line. Key kinetic parameters of Michaelis-Menten model can be determined from intercepts and slope of this line. An illustration of Lineweaver-Burke plot is shown in this figure. Here, the y-intercept of this plot corresponds to 1 over Vmax, and the slope gives the ratio of the Michaelis constant to the maximum rate. The x-intercept at -1 over Km provides a direct measure of the Michaelis constant. Furthermore, the rate constant K2 can be calculated from the experimentally determined maximum rate, provided that the total concentration of enzyme is known. This rate constant is referred to as the turnover number of the enzyme. It represents the maximum number of substrate molecules that enzyme can convert into a product per unit of time. As such, it is a measure of the enzyme's catalytic activity. Note, however, that the Lineweaver-Burke plot does not provide values for the individual rate constants K1 and K-1 that are part of the Michaelis constants definition. Despite this limitation, the Lineweaver-Burke method remains an essential tool in the study of enzyme kinetics. It offers insights into the catalytic efficiency and behavior of enzymes under various conditions.